We're here today to hear AnVIL demo here from Jason Walker, and we're going to see some demonstrations on QC analysis pipeline. Jason, take it away.
Yes, thanks, Stephen. So this QC analysis pipeline was this is basically our GitHub repo, so I thought it's best to just start from the source of the information. So we'll start with our README here. As it explains, this is a WDL pipeline that implements a QC workflow for human whole genome or exome targeted sequence data. You'll see here in a bit, there's been some previous AnVIL demos and resources on this workflow. There have been some updates that includes the addition of a exome compatible workflow, which we won't be demoing today because sadly there's not a great exome data set that's just publicly available like there is for the whole genome data, where we'll use an NA12878 available CRAM file to process this QC workflow on whole genome data. So I'm Jason Walker, but I'll also introduce now Thomas Mooney. Tom will be walking us through how to use the AnVIL platform to perform this data processing work. This was developed out of a working group that was early part of AnVIL that actually overlapped with the Centers for Common Disease Genomics. So the acronym there being CCDG that I'll use moving forward. Let me just move some windows around here just real quickly. Sorry about that. So just to briefly highlight CCDG, this presentation is not necessarily about CCDG or the CCDG data. There's actually a paper that'll be released hopefully here in the next year. It's being finalized at the moment. But basically, CCDG was one of the larger genomic studies. This would even predate the All of Us projects. The goal was basically to sequence over 100,000 whole genomes and 125,000 whole exomes. As part of the whole genomes, I think we actually reached 136,000, almost 137,000 whole genomes. And I believe on whole exomes, we reached 235,000. So QC was a big part of this. We really focused on the AnVIL project, and of course, in terms of scale, utilizing the cloud resources to process such a large volume of whole genome data, even for QC purposes. There are many cohorts that are part of CCDG. You can find a lot of those through the data portal and the data catalog, actually. The data catalog and the data explorer on the AnVIL page through the AnVIL portal. There's cardiovascular disease studies, many other types of studies here related to diabetes and autism, Alzheimer's, neuropsychiatric diseases. So without going in too much more into the actual CCDG project, I wanted to hop over to the AnVIL portal. This is the landing page for the AnVIL project. So if we go to data sets, just to show you an example of the data catalog, and if you were to drop down under the consortiums, you would find CCDG. And of course, as part of the CCDG data, we have both exome, also arrays. We're going to look at the whole genome data here, and you would need to apply for access to some of these studies. And if you were to click on these studies, it'll actually provide you more information about where you could apply for access. And within one of these workspaces, we're just going to hop over here to this workspace. This would be the landing page where you see the number of subjects, a lot of information about the cohort, and a lot of just background information. Specifically, here we see that these are GRCH38 CRAM and VCF files that have been uploaded for this cohort as a part of this. So for all of these cohorts, we have executed or run this QC workflow, generated the output for all of the CCDG data sets. Again, in the data catalog, you will only see the released data sets. There actually are many more to be released. I'll kind of mention that in a minute. But more specifically, Tom will go into how all this data was generated, but I wanted to give you a glimpse of what the data looks like for some of the CCDG cohorts. And specifically, this cohort was analyzed with a threshold file, which Tom will also go into a little bit of detail, but the cutoffs were 19.5x coverage for mean coverage. And you can see many of these are above that, but our first sample here is only 18.9. There are many other QC metrics, but that was one of the main pass-fail thresholds. And so then we can see, of course, we could filter and query for all of the data that meets these thresholds that were defined by the CCDG consortium. So that's kind of a high-level overview where we're at today with CCDG. Just wanted to briefly pop in a slide here. So this is just a summary of the manifest of CCDG data, which was just recently updated in June of 2024. This workflow, I mentioned it was updated to work with exome data, but in addition to that, we added a sex estimation from the sequence data using this method called RX. We needed to compare the RX values of this estimated sex value from the sequence data against the known phenotypes. And as you can see, just back in November, the phenotype information that was available, we actually had only mostly like 70% of the whole genome data was unknown or unspecified. After working with the CCDG consortium and the AnVIL project, we were able to resolve all of the organization of the data in such a way to correctly resolve those values for now almost 80 or over 80%. So there's only 17% that are unknown or unspecified. Again, that's all kind of in the works, and there will be a CCDG paper that kind of goes over the data. That's also in the works, but we're just going to focus mostly on the workflow. So I also wanted to highlight the help section, which you should probably be familiar with if you landed here in the AnVIL demos. The AnVIL demos is a part of this, and there's many events listed. And this help section includes the AnVIL support forum, help desk, and there's many other resources here. But if we hop over to the support forum, there's lots of discussions, including discussions of how to run these workflows. And I see Fred is here today. I just wanted to point out a lot of work. We don't want to duplicate the effort here. There's been a whole section on scaling with workflows that goes over a lot of what we're going to discuss today, although today's section is a little bit updated. This includes, you'll see a little bit of how you could turn on the exome version of the workflow, but also some of the RX sex estimate results, which are new to the workflow. And I'll just quickly highlight that there's slide resources out there from Fred and others that worked on this project before, and highlight that there's links to another YouTube video of Fred from last year in June 2023. It's a great resource. This actually goes over not only running the workflow, but how to combine workspaces from different data sets like the HPRC Pangenome project, as well as the 1,000 Genomes workspace. So we won't necessarily go over the details of how to combine those workspaces, but we will show you how to run this QC workflow today. There's the AnVIL demos. If you just go to the search bar and search for demos, these will pop up. Right. So I mentioned the CCDG data before we dive into the workflow specifically. This is a little hard to view. It's a work in progress, but it's basically showing you the RX estimates with those error bars. But at the end of the day, this boils down to having about a 99% accuracy rate with predicting the, we have a true versus false match. And certainly some data sets are enriched for false matches, and others are true. But overall, across 136,000, we have only about a 1% error rate. All right, so back to our README. So this is where you would land if you found the QC analysis pipeline. Tom will also show you this on DocStore and talk a little bit about what DocStore is. I just wanted to focus on what this workflow was used for in the CCDG consortium. This is, again, a Whittle analysis pipeline. This actually was pulled from the original whole genome analysis pipeline that Broad had created. They'd already been optimized for cloud computing. And then we pulled out many of the QC steps and added a few that were specific to the CCDG consortium. And one of the big outputs, and actually an early output of the CCDG consortium, was the functional equivalence definition. And I'm not going to go into that detail, but it's linked here what functional equivalence is. There's actually a workspace that'll run a functionally equivalent workflow, but more or less that you would, to avoid batch effects, you have an equivalent workflow that could be run across multiple centers. I don't want to list the centers because I'll probably leave one out, but there were 3 or 4, I believe 4 centers that were involved in the sequencing of the CCDG data. So we needed to make sure that everything was analyzed in the same way. So we're starting with this approved read level data. That's the data generated from each institution. This data was ingested into the AnVIL. All of it was functionally equivalent because the centers agreed on their standardized pipelines. So this really would just be a suggestion for other consortia to have a committee to determine, whether the analysis that you have performed through the data so far it meets any sort of functional equivalent standard, and how to deal with that. So all of our data went through this QC workflow. I just showed the pass-fail status, which is kind of the high-level summary. And that's ingested back into the AnVIL Terra data tables, which serve as the metadata database. This workflow, there's several inputs that are required, including the sample identifier, the CRAM or BAM file, the mode of whole genome or exome, and of course, some of your standard inputs for bioinformatics workflows like the reference and intervals and contamination sites to evaluate contamination. Here are some of those steps with some estimates, although these estimates were actually for a 60x genome. So what we've seen on 25 to 30x whole genomes is in the neighborhood of about 50 cents for the entire workflow. All right. So this goes into more detail of those required files, some of the software versions that are used. These are some standard bioinformatics tools in terms of Picard, SAM tools, verify BAM ID. We also standardized on versions that were used for some of the CCDG pipelines. So some of them may be a little dated on first pass or first look, but I believe those tools are still working correctly. I mentioned the RX identifier. If anybody's interested in learning more, there's a link out to both 2 methods, not both, but 2 different methods, RY and RX. We decided to use RX as it seemed to be more stable to fluctuations in coverage. And with any large consortium project, but also for serving as a public resource, it's hard to predict what kind of coverage levels you might estimate from the data. So we ended up moving forward with the RX value. In fact, there's even a link to the original R code that is from the publication. So there's a few notes here just about how to use and other resources. Also, this is totally open source, and we're building on the existing BSD3 license from the Broad Institute. So here we do go into a little more detail of what the CCDG data submission process looked like. And we ran this WDL workflow on all the data and came up with the pass-fail status. This table gets into some detail on exactly what metrics were collected, including mean coverage, description of that, this threshold, and the source being the Picard Collect WGS metrics tool. All right, so without going into too much more detail, these really cover the steps of running the workflow. As I mentioned, I'm going to skip to the bottom just to mention there's a YouTube video here that talks about running QC pipelines and specifically using DocStore to do so. And I mentioned the video earlier that's from last year at the AnVIL demos. And here's a link to the YouTube video, how to combine data across workspaces. With that said, Tom Mooney, do you want to maybe take over the screen sharing and start going through a demo?
Sure. That's that. All right. So just to start from the same README doc that he was scrolling through, he scrolled past this link here that says there's a text walkthrough for running WGS processing in AnVIL. And that's what I'm going to be walking through today. So, we're assuming that you already have an account set up and are ready to go with using AnVIL for this workflow. So the first step is to just go to the main page. And then we're going to go to workspaces and we're going to create a new workspace for this demo. AnVIL demo 2024 620 QC pipeline. Sure. Billing project. And description, running the QC. All right, so you do that and it will create a workspace. Take a second here, figures things out and sets up all of the permissions, all of that good stuff. And here we are. Okay, so now we can add some data to our workspace. So go over to the data tab and click import data, and I'm going to upload a TSV. So for this demo, we're running on exactly one sample. It's very simple. So we're just going to copy and paste our TSV file in here. If you had a lot more information, you'd probably upload it from a file. And so it was very quick. And here's our extremely simple sample table. We have the name of the sample. We have a link to the CRAM file. And because this is just a demo, we're pointing to a public file instead of having one that's uploaded in our workspace. If you were running this for real, you'd probably either clone an existing workspace or you would actually upload your data here through a real ingest process. And just so the table looks a little more full, it is in fact WGS data. So I added this column that says that that's true. And the last thing we're going to do here is take this thresholds TSV file that is in the repository that just has a default set of thresholds for passing QC. So you can see the, like Jason mentioned earlier, the mean coverage needs to be greater than 30. And there's few, at least 95% needs to be 10x and so on.
And Tom here, it's important to for those viewers that these are customizable thresholds, and that's one of the features of this QC workflow, is it's intended to be a resource, and it was used for CCDG, and there were specific thresholds used for CCDG, but the idea is that our community can add additional metrics or tools, but that this serves as a framework for how to generate a consortia level a pass-fail status for QC, sort of a summary with collecting all of these detailed metrics and visualizing them in AnVIL.
Yes, and so there is a separate doc page on this workflow for the thresholds that describes the format for the threshold file and then lists the many metrics that you could use and set threshold for QC. It's basically anything that comes out of the Picard tools that were run and verify BAM ID and etc. Oh, so we have this. We need to download this file. So I'll just save it to my downloads. And then we will upload it to the workspace by clicking on files, choosing upload, going back to my downloads and finding, well, okay. And so now that file is there for us to be able to use in the workflow. And so having set up the data, now we need to attach a workflow to this. So DocStore is an excellent resource for finding workflows, and our workflow is also there. And so this is the page for that. It has a copy of the QC and the README, and has this handy set of buttons, and one of them is the AnVIL. So I can click on that and find my workspace. It was AnVIL demo 2024, or this one. And I can click import. And we have a new tab here where it is. So I'm just going to move this over to this side so that we can go back to the README. Okay, so we have selected the workspace, and now we're on the workflow tab. When, like it says, we want to run with inputs defined by the data table. We're going to choose our data, one and only data table, the sample, and we will select the data. We're going to run it on everything. So I'll click the select all box, even though that's only one thing. And now we need to define all of our inputs. You can see the workflow has a lot of required inputs that are mostly like the files used for the basis of the QC. We also have the input BAMR cram itself, the is WGS that was that other column in our sample table, and the sample name. And so the repository here comes with a complete functional set of inputs that you can use. It actually uses the same cram file that we're using for this demonstration, but we're pulling it from the data table in the AnVIL just to demonstrate that feature. If you were to just select run workflow with inputs defined by file paths here on the right side, you could upload this as is, and it would just run. So we need to download this one. So save that. And then here we can click upload JSON, and and you would find the one that you downloaded. This was single sample qc.json, and it populates all of these inputs for you. You got the interval lists and contamination files and all of these things, the reference sequence that would be the same for the entire data set. But the next thing we're going to do is to switch the things that would vary per sample. So instead of hard coding this value, we will take the sample ID that's from our sample table. So this here means that it refers back to the data table we selected in step 1. And let's see, I said base name and input BAM. And even though it's called input BAM, it will accept either a BAM or a cram. So we'll use this .cram, and then we'll take this value. In actual fact, if you were going to run on exome, you would probably pick some different other inputs, like the coverage interval list. But if you did want to mix WGS and exome with the same one, you could do it this way. And then the evaluation thresholds is the last one to change. And you'll notice that this one points to an HTTPS URL in the default input JSON that we provide, but that doesn't work as well with Cromwell on AnVIL. So we're pointing it to the one we uploaded to our bucket instead. And now that we have the inputs, the important thing to do is save our changes so that we don't lose all of the work we just did. And now we move on to the outputs tab. And by default, it is going to write all of the outputs from the workflow to the same table that we're running on, which is exactly what we want here. if this weren't filled in or if we cleared it, we could click this use defaults button and it would fill it all back in for us. And we're going to delete our intermediate outputs because we don't need to keep all of that later. It'll help us save disk space in the future. And then I will click the run button and I'm ready to go. I'll see only launching one analysis. If that said 100, I would be scared that I screwed something up and go back and check over it, but it says 1. And so running leaving a little note for myself so that I know why I ran this later if I need to look back. And I'll click launch. And it will begin. It's queued up to run, and the once it starts moving, there will be additional information here for how to track its progress. Now, this workflow on this sample will take several hours to run, so we're not going to sit here and watch it. But I have another workspace where we already ran this sample and it already succeeded. And then you can see that there's these links over here that I mentioned would exist. So if I click the workflow dashboard, see all the steps succeeded. As it was going along, there was this one retryable failure. So this workflow does use preemptible instances to save money on running, but that means occasionally your job gets interrupted as it's going along and has to try again. But it succeeded on the second time. Now you may notice that call caching was enabled, and this was a second run of this particular workflow. So most of these had a cache hit and ran in a matter of a second instead of taking a long time. But if we go back, so if I were to have clicked on the job manager and authenticated to that, you get a different view of the same thing where you can see a few of the details if you like that version of the page better. But so then having that workflow completed, if we go back to the data tab and go to our sample, we'll see that there are a lot more columns in this table. As Jason mentioned, the big thing we're looking for is the overall evaluation. Did we pass or fail? This also includes more detailed information. If I scroll back to the evaluated metrics and I look at that, you can see that it provides a pass-fail status for each individual metric that was in our threshold file. So if it were to fail, you could open this and see which one that it failed. And so that is also available as a TSV file. And so if you needed it in file form, it's here. And AnVIL hopefully gives you a preview of the file if it's small, and then ways to download it. And that's similar for all the other metrics files here. There's also like one of the QCs generates this PDF of the GC bias report. So I can go through and click on that and then click through the cloud storage and open that and take a look. There's no way to show this PDF in the data table. So, and then after we've run the workflow on this and we have this data, there is also a QC notebook, multi-QC in a Jupyter notebook that can be run that will provide a very nice report from all of the QC metrics. And we have added a module to it to also show information for the RX. So the next step for setting that up would be to go over to analyses. For a brief second here, I'm going to go back to our original workspace that we ran on just to show. We go to environment configuration. So we're going to analyses. sorry, I'm going to walk through this. We go to analyses and press the start button and select Jupyter. Oh, right. Download the notebook. So we download the notebook like we did the other files. Save, downloads. Great. And click here and downloads. We've got multi-QC. Open that. And now we have the multi-QC notebook here. And click on that. And then we don't have an environment configured yet. So when I click open, instead of launching an environment, it's going to ask me about the things that setting it up. We have, as I mentioned, a custom version of multi-QC based on a fork that the Broad made that can read from buckets in Terra and AnVIL. And we've modified it further to support the RX. So this script, I can show it to you very quickly. All it does is install this fork of multi-QC. And so multi-QC, if you're running on a real data set, requires a bit of resources. So we just bump those up normally to 2 and 13. And then auto pause is very important. So just noting that it's there. And then to set this up, I would click create and it would launch the environment. But since this is the one we didn't, it is still running the workflow. We'll go back to our other workspace instead. And in this one, we've magically already got the environment started. And so then when I click open, we'll open the notebook. And we just wait for it. Kernel starting. Just waiting. Being patient. So the top of this notebook mentions the same thing again about this startup script and has a copy of it here. And if you can't use the startup script, there is this backup way to do it, but we don't need to do that. So we will move on. And now that the notebook is ready, we can run the 2 cells to do our multi-QC run. There's just this one, so I can run that. And then I can move down to this next one, and this one does all the work. If you look at the code, it looks for files that multi-QC understands, and then we'll run multi-QC and save the report. So if I click that and run, it gives us a note of its progress. If this were run on a large data set, it would take a bit longer, but since this is a single sample, it's fast enough to show right now. And it found our output files in the bucket. It's seeing which ones multi-QC understands and downloads those metrics to this machine to run multi-QC, and then uploads the report to the bucket. So at the very end, we get this link and it takes us to the Google Cloud page, and I can click the authenticated URL. And we have our freshly generated multi-QC report. You can see it was generated right now. Have the summary. The RX tables are not very exciting with one sample. This one sample is XX, so it's 100% that. And then we have the other multi-QC reports on the various QC things, other QC programs that were run. And just as an example, we have, we also, of course, my session signed out. if I reload, so we previously ran this on one of the 1,000 genomes data sets. And so that also has a multi-QC report, and this is what it looks like with a bit more samples. So this one is just like each sample and its RX score, and then colored by the reported sex. And then we have the breakdown in this data set. It was roughly a 50-50 split. And then you can see that most of the time it gives a definitive result, but for 18 of the samples out of these 3,000 and some, it did not fully commit to one answer or the other. And that is my walkthrough of this walkthrough.
Thanks, Tom. Yeah, so we're welcome to take any questions. Again, I think the summary here is this is just demonstrating the capabilities of performing a QC at a large volume across many different data sets in cohorts as part of the CCDG project. But now those QC resources are available to the community. We welcome pull requests to our GitHub site. You can find us on DocStore as well. It's part of the AnVIL organization on DocStore. And yeah, we're welcome to input and working with other consortia and or collaborations or organizations that would look to standardize WGS QC. At this time, we'll take some questions.
Thank you, Jason. Thank you, Thomas. I really appreciate your work here.